Hello and welcome to a special video. Someone asked me if I could do a video on doing a natural exam paper for F212 and I went okay. So this is just mainly so you can see what the answers need to be like. What I would suggest is that before each question, there are 6. Before each question I will leave a slight gap so you can pause the video, do the questions. I'll link the paper and the mark scheme in the description, but you try each question, then watch and see what the recommended answer would be and how you answer it. So quick little pause for question 1. Good. So question 1. Oh, a lot of reading for me. Yay. Milk is considered to be a complete food containing most of the components of a balanced diet. A student carried out a series of food tests on a sample of milk. The student's observations and conclusions are drawn in table 1.1. Complete table 1.1 by naming the molecule being tested for, stating whether this molecule is present or absent. So firstly, you see food tests. So the first row has been done for you. Benedict solution, observation brick red precipitate. If it's a Benedict solution, it must be a reducing sugar, and since it's got a precipitate and it's quite a dark coloured one, it must be present. Biuret 1 and 2, so that's a protein, and lilac colour means it is present. Iodine solution, so that's starch, and yellow brown absent. You would expect blue black if it was present. Although the student entered present for the lipid in the first row of the table, he was unsure whether the result was correct. Suggest why the student was unsure if the positive result for the lipid was correct for the milk sample. Very simply, milk is white. If the observation for a lipid is a white emulsion and the milk's already white, you can't tell if an emulsion has formed. Triglycerides are a type of lipid found in milk. Describe the structure of a triglyceride molecule. Now you can get all 3 marks by just drawing it, but I always suggest write it as well, just don't conflict yourself. 1 glycerol molecule, 3 fatty acid chains, 3 ester bonds between the glycerol and the fatty acid. Simple. State 3 roles of lipids in living organisms. Now this is one of the questions where there are about 10 answers, but he only asked for 3, so look at the mark scheme for the rest. If I haven't put your answer down here, it's probably still correct. Energy storage, waterproofing, thermal insulation. Also, I should say actually, in this, tick just shows what's given the answer. If I've underlined something in black, it means that's a keyword you must have. If I've put something in brackets, it means you don't need it. And if I've circled a red tick, it means that I've already got the maximum number of marks for that question, and any more is just extra, but are still valid marks. Suggest one difference between human populations with diets high in animal fats have a lower life expectancy than those with diets high in vegetable oils. Suggest one difference between lipids from animals and those from plants. Animal fats are saturated, plant fats are unsaturated. You only need to say one or the other. Here's a graph. Animal fats are thought to raise blood cholesterol levels. High blood cholesterol levels can lead to premature death. Figure 1.1 shows the relationship between blood cholesterol level and annual death rate per 10,000 of the population, men and women. Very common. You'll get one of these questions. You will get one. Describe the trends shown in figure 1.1. Death rate is greater for men on average. Death rate of both genders rise with increasing blood cholesterol. Death rate for men initially falls. And men increased rapidly from 32,000 deaths at 6.7 mmol dm-3 to 60,000 deaths at 6.2 mmol dm-3. Always have, in any answer where it says describe the trends, always quote data. That's a mark. Increased blood cholesterol levels are associated with certain medical conditions. Suggest 2 conditions that may be associated with increased blood cholesterol. Coronary heart disease, atherosclerosis, there's a lot. So yet again, look at the answers. Okay, and that's question 1. What I would like is if you can
do each question, and then once you've finished all of them, post what you got overall in the paper and what you got on each individual question. You know, what's your weakest topic. So, you know, I mean, I've got 137 subs now, or a bit less maybe. We can all, you know, work together to help revise. It helps. So for a few seconds to do the pause for the next question. Good. So, when a new species is discovered, it needs to be classified. Define the term classification. They give you so much space for this. Grouping organisms into taxa based on their similarities and differences. 2 marks. Suggest what criteria taxonomists may take into account when classifying a new species. Morphology and anatomy, biochemistry, genetics, behaviour and physiology, also phylogeny, that as well. Place letters representing the taxonomical groups in the correct order. The first one's been done for you. So the order is kingdom, phylum, class, order, family, genus, species. So it must be V, T, S, R, W, U, Q. You get 1 mark for each 2 you get in the correct order. Right. The classification of organisms into domains is relatively new. Describe the differences between a classification system based on domains and one based on kingdoms. Right, you get 1 mark for saying there are 3 kingdoms and 5 domains. I've split it up just a bit easier, but you must say 3 kingdoms and 5 domains. The 3 kingdoms are Eubacteria, Archaeobacteria and Eukarya. And the 5 kingdoms are Plantae, Animalia, Fungi, Protoctist and Prokaryote. All eukaryotes are placed in the same domain, Eukaryote. All prokaryotes are in the same kingdom but split into 2 domains. And they are classified on rRNA, that's ribosomal RNA, membrane structure, particularly lipids, and protein synthesis. So a very quick 12-mark question there. This is probably one, I think, say actually the easiest one on the paper, so you don't want to lose many marks. It's mainly just definitions. So a few seconds to pause. Good. Next question. In Scotland, this is the most boring question on the paper for me personally. In Scotland in 2007, there was a major food poisoning outbreak that killed 3 people. Suggest one group in the population that is likely to die from food poisoning and give a reason for your suggestion. There's the young, elderly, HIV infected, anorexics, and same reason for all. They all have a weakened immune system. The food poisoning outbreak involved the bacteria Escherichia coli 0157, E. coli 0157, which has been responsible for contaminating meat products. The meat had been stored at 11 degrees Celsius rather than the recommended 5 degrees Celsius, and this led to meat spoilage. Explain how bacteria cause food spoilage. Bacteria divide and replicate. They secrete enzymes which cause food to be digested. Then the byproducts of this digestion cause secretion of toxins and waste products that cause a change in the appearance, smell, texture and taste of food. Food normally spoils much faster if stored at temperatures higher than 5 degrees. Explain why. Bacteria replicate faster, so more bacteria are present, so more toxins and waste products produced. So you need to say that or the other one. You can say both, but usually go for one or the other. More enzymes are secreted, which are more effective at higher temperatures. Both enzyme substrate molecules have more kinetic energy, so more enzyme substrate molecules are formed, rate of reaction increases. Food can be preserved by keeping it at low temperature in a refrigerator or freezer. Name 2 methods of food preservation. Now there are about 10, so please look at the mark scheme. I've just chosen my 2 favourites. Pickling, the low pH denatures the tertiary structure of the enzymes. Cooking, denatures the tertiary structure of enzymes, same reason. 4 marks. And then the first big question of the paper. Microorganisms such as the fungus Fusarium can be grown and then purified to produce mycoprotein. This mycoprotein can be used as a food source for humans. You know, vegetarian. I'm vegetarian myself, so I've eaten it. Here's a table. Use the data in table 3.1 to describe and explain the advantages and disadvantages of using microorganisms to produce food for human consumption. In your answer, you should make comparisons with the information in table 3.1. You get a quality of written communication mark, that's 1 mark, if you have compared data in the table twice. I just do it for every single one, to be honest. It doesn't take that much more time. So, advantages. Mycoprotein has less energy than beef, given a figure. So it's useful for weight control, or slimming. There's less total fat and less saturated fat, therefore less cholesterol, so a low risk of coronary heart disease, atherosclerosis, or the others, stroke. Contains carbohydrates. Disadvantages: less iron, 0.1 milligrams compared to 2.6, so risk of anemia, and there's also less protein. And that's 20 marks there. I say this is not the hardest, but you know, I think it'd be very easy to skip over this when revising. Luckily though, this came up in January 2013, so I doubt we'll get a lot on it in June. Hopefully. I don't want it. So, pause the next question. Good, right. DNA and RNA. This hasn't come up for a while. It could come up in June. I hope so. This is a nice, easy topic. Figure 4.1 is a drawing that represents molecules of DNA and mRNA. The mRNA molecule is shorter than DNA molecule. State, using only the information in Figure 4.1, one other way to distinguish between DNA and mRNA. mRNA is a single strand, DNA is a double strand, mRNA isn't a helix, DNA is. Give one further difference in structure between DNA and RNA. RNA contains uracil, DNA contains thymine. RNA contains ribose, DNA contains deoxyribose. DNA and mRNA are both involved in protein synthesis. The mRNA molecule carries the code for a protein, leaves the nucleus and attaches to a ribosome. The ribosome is the site where a protein molecule is formed. Complete the following statement. A sequence of DNA nucleotides that codes for a protein is a gene. Suggest why DNA is not able to leave the nucleus. DNA is too big to fit through the nucleus pore. Explain why the mRNA molecule is shorter than the DNA molecule. Very easy, just to lose 1 mark here. mRNA only copies 1 gene. DNA has many genes. State the obvious. Enzymes evolved in the production of mRNA in eukaryotic cells. One enzyme is inhibited by a toxin, alpha-amanitin. Figure 4.2 shows the effect when alpha-amanitin attaches to this enzyme. Sorry for any pronunciation. It will be Amanitin, maybe. Explain how alpha-amanitin stops the formation of an enzyme substrate molecule during RNA production. Simple way to just do this. Look at where it is. We know it's inhibiting, so because it's preventing something happening, so it's inhibiting. If it's at the active site, it's competitive. If it's away from it, it's non-competitive. So alpha-amanitin is a non-competitive inhibitor. It fits into an area away from the active site, thus changing the shape of the active, changes the shape to changes the tertiary 3D structure of the enzyme, which then also in turn changes the active site shape. So the substrate no longer is complementary to the active site. Must say to the active site. Obvious, I know, but you need to say it. The Roman Emperor Claudius was poisoned by his wife, Agrippina, when she gave him death cap fungus to eat. The death cap fungus contains alpha-amanitin. Suggest how the toxin alpha-amanitin may lead to the death of an organism. It inhibits mRNA production, preventing protein synthesis. And now, you know, we're over halfway through the paper. We get a very simple few marks now, just to relax a bit. Enzymes are globular proteins with a specific 3-dimensional shape. The shape is determined by the primary structure. State the meaning of the term primary structure. Sequence of amino acids. And now name the bonds labelled A, B and C. A is between R- and R+, which means it's a positive R group and a negative R group must be ionic. Next one's between O and H. Hydrogen bonds form between hydrogen and oxygen, nitrogen or fluorine only. So since it's hydrogen and oxygen, must be hydrogen bonds. And name the bonds labelled C. Sulfur to sulfur must be disulfide. When proteins are heated to a high temperature, their tertiary structure is disrupted. Explain how this occurs. This increases the kinetic energy of the enzyme, causing it to vibrate. This breaks the weaker hydrogen ionic bonds, also hydrophobic and hydrophilic interactions, changing the 3D structure of the enzyme, denaturing it. 17 marks there. Pause for the next question. You know, 2/3 of the way through the paper is going well. Good. So 5. Smoking increases the risk of lung infections. Explain how the mucus and cilia in the air passages reduce the chance of developing lung infections. The mucus tracks bacteria, viruses, pathogens, dust. The cilia waft the mucus to the back of the throat where it is swallowed and digested. Here's a nice diagram. In an individual with bronchitis, the mucus contains a large number of pathogenic bacteria. Phagocytic white blood cells can destroy the bacteria. Figure 5.1 shows a sequence of events that result in the destruction of a bacterium. Describe the events taking place at stages A, B, C and D. A. Phagocyte binds to a pathogen and a receptor binds to an antigen. Just explaining the first point there. B. Bacteria engulfed by endocytosis or phagocytosis. A phagocytic vacuole is formed. Now in the mark scheme it doesn't say phagocytic vesicle, but I think that would be acceptable. Lysosomes at C fuse with phagocytic vacuole, secrete hydrolytic enzymes which digest the bacteria. And D. The bacteria is absorbed, or the broken up bacteria is absorbed into the cytoplasm by diffusion or active transport. The immune system will produce specific antibodies in response to infection. Name the type of cell that produces antibodies. Plasma cells. Now, you might be allowed B lymphocyte, but B lymphocytes they will change into plasma cells which secrete antibodies. And the other big question, which I didn't even need half the page for, describe how the structure of an antibody molecule is related to its function. In your answer you must clearly link structure and function. So here you get quality written communication mark for twice, linking the structure with the function, but do it every time. Y-shaped molecule with 4 polypeptide chains, basic description. 2 short chain polypeptide chains, 2 long chain polypeptides. There's a constant region, so the antibody can bind to the phagocyte. It's the same for all antibodies. There's a variable region which provides specificity as it has a complementary shape to the antigen. And there's a hinge region which provides flexibility. Now there's a few more you can put, but that's all you need. You don't need any more. And another simple mark. Identify the type of immunity provided by antibodies in breast milk. So it's breast milk. You're being given breast milk and that's natural. You're not being injected with anything. It's natural. So we know it's natural active or natural passive, and you're being given antibodies. You're not being given the disease to produce your own antibodies, so it's passive. So you now pause to the next question. Last one. Good. Right, now there's hardly been anything on biodiversity in this topic, so here's the question. Before any major development can take place, an EIA needs to be carried out. One such development is the proposed extension to the M27 motorway in Hampshire. This extension would cut through an important heathland ecosystem. Suggest 3 aspects of this development that would need to be considered when carrying out the EIA. The biodiversity, rare and endangered species, rarity of the habitat, and if a reduction in the size of the habitat would occur. There are a few more as well. An ecologist carried out a survey of butterfly species on the heathland. The ecologist walked along a marked path on 4 different days in June. She counted the number of butterfly species and the number of individual butterflies of each species. She says how a method of collecting data could be improved. Well, the obvious one actually is that she's just counting, she's not collecting them. So use a sweep net. Collect a sample from a wide area. She's just going down a little path. Collect at different times of year and day to get a more accurate representation of the whole time scale, and do mark, release, recapture, so you prevent recounting. Right. The ecologist results are shown in table 6.1. These results can be used to calculate the Simpson's index of diversity for butterflies in heathland. Blah 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 blah. Now, in the actual exam there was an erratum notice saying that 0.09370 was wrong. It's a slightly different number. So these are the numbers if you use the correct value, not the mistaken one. But if you just look in the mark scheme, will give you the one if you do use the incorrect value. Sorry. Suggest the implications of a high value of the Simpson index of diversity on planning decisions. High species richness, so the area should not be developed. Then we have a dichotomous key. You can see the letters there. I'm not going to go through the whole thing. You can do it yourself. And finally, State why small heath and large heath butterflies both share part of their scientific name. They have the same genus name, mark, because they have similar characteristics and share a common ancestor. And that's it. So 20 minutes to go through that paper, and we're done. So yeah, post what score you got on individual questions, and overall, if there's anything you want help with, feel free to email me, munrot07@yahoo.co.uk, message me on the student room, or leave comments or inbox my YouTube, whatever you want. Yeah, and I hope that helped. So yeah, rate, subscribe, like, comment, whatever. Yeah, hope you enjoyed this, hope this helped, and goodbye.